No, está grabando ya.
Así es.
Voy a compartir mi pantalla.
People will be joining during the session because of there's a lot of people that is joining from Ecuador, but Ecuador is 2 hours behind us. So maybe they're just waking up.
Yes, it is.
All right.
Good morning.
Good
You know,
morning, Rodrigo. Okay, so I'm going to, there you go.
Okay, this is my
So a
presentation.
few words just to leave those words here in the recording. This is our third session in our webinars that we are preparing for this that we call graduate workshop. This is we are trying to show what are we doing in our SDG project that we are doing together with uh, University of the Life Science from Norway, from the Bursa of the Lag University from Turkey, and from Universidad Católica del Norte from Chile. So we have a 4-year project that each institution is sharing what they are doing in terms of aquaculture and agriculture sustainability. A little explanation is our partners from Norway, they are focusing in use agriculture feeds or debris or waste coming from that industry, how to use it into the aquaculture industry. So doctor people from Norway are doing that, and they did their lectures, I guess, 2 months ago, and those are available on our webpage for you to see. You were unable to see the live speaking. Then our colleagues from Bursa of the Lag University, they are not aquaculture researchers, but what they do, they are looking how to produce energy from waste coming from the aquaculture or from the agriculture or from any other kind of industry. So they were also talking about that. And the Universidad Católica del Norte, the researchers are focusing in aquaculture and aquaponics. So we are, in that terms, we are domesticating some kinds of animals that we have here, locals, like the rainbow trout, that is common between Norway, Turkey, and Chile. But also we are working with a local uh, freshwater prawn that Dr. Cristina is going to talk here. And basically that it is. Next year we will see what we are doing, but for now this will be our last seminar for the year. So whenever you are ready, Maria Cristina, you can start.
Okay, thank you. Good morning for everyone. Today we will share some principles of and methodologies for crustacean production and that we have developed in Chile. First, we need to know two aspects. The management of captive breeding in the production of juveniles of crustacean from both freshwater and marine water. And this work also considers condition indicators that allow us to evaluate the development of a culture and predict, for example, whether the juvenile stage will be reached and which stages are critical and which stages are less conservative and can be optimized. Crustacean farming in Chile is important because we have a connection with the native species and to the natural and cultural environments of freshwater species that have been important even in indigenous culture. We have here different kind of pictures. Some pictures belong to the Inca period, and for this reason it's very important for us. Our aquaculture is development in a small scale in arid zones where the water is the most important resource. And for this reason, aquaculture involves the use of integrated system and water recirculation. For us, it's very important, this work. We begin this research since 20 years or more, and it's very important for the local aquaculture. The juvenile production involves 3 methods for obtain juvenile shrimps mainly. Each method have advantage and disadvantage. So it's possible to use one or 2 methods in combination to optimize the production of shrimp that will be starting a point for commercial size shrimp. In the photograph in the left, we can observe first the capture of juveniles shrimp from their natural habitat. We will see a beach, and the fisherman walk along the beach catching juveniles for target species. In this case is Lithopeneus banamei, a tropical shrimp with their nets. It's a basic method for catching the shrimp and have one advantage. Is the low cost, but one disadvantage. Not only is the target species caught, but also we have other organisms and juvenile of other crustaceans. For this reason, it's very difficult to identify the correct species. In this case, Lithopeneus banamei. And then how we can to be sure that we need the correct species. And for example, how is the quantity that we caught the Lithopeneus banamei? In this case, only we have a percentage represented that touches species. It's very difficult to be sure that we have 7% of Lithopeneus banamei. For this reason, when the fisherman commercialize the juveniles in the border of the beach, we only have a percentage of the representative species. It's very important, this method, because the juveniles have a high quantity in the capture. In the photo that we have in the middle of this slide, we have the capture of females with eggs or with spermatophore. This alternative is very important because we have sure that the species is the correct that we need to capture. Each female represent one family of juveniles, but we don't know who is the father, or this female could be reproduced with multiple males. We don't know the real traceability for to know the father, but certainly each female is a new family. In the third option on the right is to obtain juveniles thought captive breeding programs. It's possible select breeding individuals, create families, maintain the traceability, and implement a reproductive control plan. In Chile, we use the second and third method. For us, it's impossible to use the first method because we work with freshwater prawn that we obtain in the river. For this reason, we prefer use the second method with females with eggs or juveniles from the laboratory. Working with the reproductive organism is easy when you can differentiate males from females when the sexual dimorphism is present. We can identify then by their primary or secondary sexual characteristics. In the capod crustacean, the genital pore in males is located at the base of the fifth pair of periopods, and in females, in the base of the third pair. In this picture, in the left, we have a female with the genital pore, and in the right, we have the male that the genital pore have in the fifth pair of the periopods. The picture on the right, we have our local species. The name is Criphops cementarius, but it's a paleomonid shrimp, freshwater shrimp, and we have an important differences between male and female. The male is a bigger and have the second pair of the periopods more development, and the female is smaller and have a wide abdominal part, because in this zone the female carrying the eggs. In this picture, we can see the same situation, but in crabs. We show the secondary sexual characteristics and have a more part in the pleopods, with the first pair is modified for copulatory appendix, but the gonopore or genital gonopore is located in the same part the periopods. Respected with the gonadal development, it's very easy to recognize the stage of maturity of the gonadal. We have here in the left sequences of the gonadal maturation of since stage with the gonad more development, and in the immature stage. This is very important because in aquaculture we use many kind of indicators that provide the quickly information for identify the reproductive stage. We need to more studies, for example, histological analysis, but with this information, we translate the indicator for characteristic that we need to observe quickly. In the case of this shrimp, for example, the gonad, the position and the development of the gonad, we will see very easy in the abdominal section for the females. In other cases, we have characteristics very important in the males. For example, we have a reproductive situation with the males when it's development one pair of appendix more than the others. This situation is because this species is Clipheus cementarius, a freshwater shrimp, have a characteristic because they have a process of ecdysis for the reproduction, because it use this appendix for attract more than 1, 2, or 3 females. This development is the accord with the maturity, sexual maturity of the gonad. Here we can to the change of color is a white color in males, but in the females, the gonad is, we can see a dark brown color, and then we can see ovigerous females. The females carrying the eggs in the abdominal section. The eggs are accommodated in the pleopods. The eggs for addition, the eggs produce. For this reason, the reproduction in many crustaceans is associated very strongly with the growth, with the molting or ecdysis, and the development of secondary sexual characteristics. It's very important because the reproduction need to the most important energy that this crustacean need to prepare the process of the reproduction. Is the most critical activity when we need to control in laboratory for the obtain a good reproduction. This is the captive crustacean breeding. We can see one male, a big one male, with 2 or 3 females if you can see the appendix, the big appendix, this the second pair of the periopods, and they use for create an species of no sé cómo se dice, cerco, but a place for the females captives. The female is prepared for the reproduction when it is present anecdosis. In this picture, we can see the female and its exuvia or ecdysis, and in this moment, the male can positionate the spermatophore in the middle of the cephalothorax of the female. And after that, in 1 hour approximately, we have a female with eggs. In laboratory, we can see females with brown eggs or female with green eggs. In the river, the eggs are brown, but in the laboratory, we can see different colors. It's not a problem, this, because depend of the food and the tank color and the water, the quality of water, and change the color of the eggs. But the larvae is the same quality. Here is a video. We can see the process of mating with a big male. This arm, the big arm, is very delicate, because when they lose this arm, need to more energy for repair that loss, and probably the other arms begin to grow. For this reason, we have crustacean that have a right calliped bigger than the other. When we have eggs, we need again other indication. Why? We need to check the development of the eggs, and with this information, we separate the females that having the same embryonic development. We put the females in these tanks with the same embryonic development. We check the stage with a little sample. We observe in the microscope and select the females depend of the embryonic development of eggs. We have or determine 5 stages: unfertilized eggs, and the eggs that have an embryo. We only use 4 stages. Is the most important stage only for aquaculture indicators. Probably in the real development of the eggs have more stages, 7, 8, because the changes are a little, with a little step, we need to see for the aquaculture characteristic that we need to see quickly. For this reason, we have this indicator. The days between one stage or other is about 4 days or 5 days up in the time. In the example with crabs, this is a marine crab, it's the same. We have a female with the eggs in the abdomen, and we have the initial stage of the eggs and the final in the right stage of development of the embryo. And after that, in one day, in the next day, we have larvae. It's a high probability that we had larvae. This is our laboratory of production. We work with the crustacean of freshwater crustacean and marine crustacean, because our laboratory have two type of water. We work near to the sea, and we use the fresh water from the public system, and we need to prepare the fresh water because we need to eliminate the chlorine, and we need to use water with filter to 1 micron and use the UV system. We use alternative or complementary cultures with microalgae, rotifers, and artemia, an important live food culture. Is very important for us, these 3 type of organisms, because it's the base for the food plan in the first stage of the larvae. In this slide, I present to you a live food and general feeding plan with 2 type of food, live food and formulated feed. This is the quantity. We start with microalgae and rotifers in this quantity of cells by mineral and rotifers. And in the second stage of soya, we start with artemia. Not in some cases, the first soya don't have the mouth development. Only in the second stage of soya, the larvae have the mouth open. And for this reason, we start with microalgae and rotifers. The size of the food is very important for the size of the mouth, the crustacean. In the fourth soya stage, we start with formulated feed ad libitum. Is a preparation that we elaborate in the laboratory with different kind of products like eggs, milk, and a, a little bit of crustacean for the chitin and a protein that we use calamar, no, se me olvidó cómo se dice calamar. ¿Ah? With the tibia or calamar. And we use a specific oil with the omega-3 that is important. We prepare, we cook this egg custard and we give to the larvae at libitum in the little pieces from between 300 or 500 micrometers, and it's the most important formulated feed because we we give the micronutrients that the larvae need in this stage, because in this stage the larvae have the stomach development and can to process the formulated feed. The environmental conditions, we have this we have these values for freshwater shrimp, crabs, and Lithopeneus banamate. That's important because this species is the most known in the aquaculture international, and the value of range of the temperature, salinity, dissolved oxygen, pH, and total ammonia. This is the value of the critical number that we need to observe. For the larvae, we have two different kind of indicators. We use we use 10 indicator because the larvae is easy to observe. It's easy to see in the microscope. It's easy to see in the tank, and because they are, we can see the external characteristic and the internal characteristic. For this reason, we use, for example, the appendix, the development of the external appendix, the spines, the gills, the transparency of the muscle, the foregut, the food in the foregut, the development of the gills, and the chromatophorus, that we have an important information about the development of the larvae, and we can to identify a critical stages, mainly in the first period of the development. We have larvae with 4, 10, or 18 stages of development. But there we have identified the critical stages because they are very conservative in this in the characteristics, and we can identify that critical stages with the with the mortality, with the high mortality, and we can discover that we have 1 or 2 or 3 maybe stages, critical stages, but always is the mortality is in these stages, nor in other. For this reason, we can reduce the mortality in these stages, but we can't change the critical stage. This is very important. It is discovered that we discovered this situation because we take a picture every day for each larvae, sample of larvae, and we can identify these 10 indicators, and we have a model of the development, the larval development, and we identify the critical stage. Here, for example, there is an important larval condition indicator. Is the beetling reserves in the photograph in the left. We can see a little balls of lipids and proteins. This is the first larvae that release of the eggs with the first zoea, and we have a high quantity of these balls, that is reserves, beetling reserves, and is a good indicator that the larvae born in very well conditions. But in the picture of the right, we can see in the middle of the cephalothoracic zone, in this larvae, the stomach and the digestive gland is empty, because these reserves are occupied by the larvae by ecdysis process for growth, for development structures, and for this reason, this larval indicator is very important. But because mark the start with the food that we need to prepare and give to the larvae, live food and formulated food. The gills in the in the picture of the left bottom is very important, because the larvae at first realize the capture of the oxygen from the water by the epidermis, not by the gills. The gills are development when the larvae are growth, and we can see a little gills that is formed, but maybe these gills are not functional in the earlier stage. The gills are functional when the larvae is a juvenile. For this reason, the quality of water is very important for the development of the gills, because we need to clean water for reduce the impact of the protozoals and organic matter that will be affect the development of the gills. Here there is another example for the crabs larvae. We always take the same picture for define one stage, another stage. When we don't know the development of the larvae, we need to take picture and identify 5 indicators that we need to know when the larvae change the stage, and we can know the days that the larvae change that the another stage of other. This is the first stage of zoea in crabs. We need to know in the first picture, we need to know the development of the spines, the abdomen in the second picture, the abdomen development and the segments. In the telson, the spine and the formulate of the spines. This is a larvae with a bilateral symmetric, it's a bilateral symmetric, and we need to count the spines, 1, 2, 3, 4, 5, right and left, and for this reason, this formula is 5 plus 5. When this characteristic change, we have the security that the larvae change the stage, because we need to know when the larvae change the stage, or it's prepared for the change of the stage. In the second stage, zoea 2, we take the same picture. Again, the cephalothoracic zone with the spine, because change in the size, the abdomen, and the telson, and we can see that the second stage don't change the spine in the telson. But we have to see when the larvae is prepared for the ecdysis. We can see in this border of the telson a double line, and this situation, we can predict that the next molt is near. The third stage of the eye is similar, but we can see, for example, in B picture, we can see the beginning when the formation of the pleopods. And in the telson, we can see a new spine that born in the middle of the telson. When some characteristic change, we can think that the larvae change of the stage. When they appear a new characteristic or some characteristic that we see, but that we saw in the first stage, and in the next stage, that characteristic disappear, we can indicate that the larvae change its stage. We check about 10 or 20 larvae by day with this picture for determine when the larvae change its stage of development. Because we need to build models of the development larvae. And we In the same line of time, we analyze the different indicators. And in this work, we can to identify the critical stages. And that model or pattern is very conservative in many species. And when you link that characteristic with the mortality, you can see that the model predict the juvenile quantity, the last survival, and you can adapt the protocols of daily work in the laboratory. When the mortality is produced in other stage, indicate operational problems. This is the fourth stage of SOEA. We can see the development of the pleopods. One pair of the spine in the telson, the development of the structure of the cephalothorax appendages, the megalopa stage in the crabs, and here we have juveniles of crabs. For this species, Omalaspis plana, we have 4 stages of the development. It's very easier. The culture is, the time is about 25 days, but in the freshwater shrimp, we have 18 stages of SOEA. It's a long time for the development, about 45 days. This freshwater Criphops cementarius is the same family of Macrobrachium. It belonged to Palemonid family, but is the one of the larvae development more longest I know. We are identified 4 stages, critical stages. The second SOEA, because in this stage, the larvae are open the mouth. In the 4 and 5 SOEA, when the larvae development the stomach. The 10th SOEA, stage of SOEA, when the larvae finish to build the structures. But when in the last stage of SOEA, in the 18th, previously to metamorphosis for juvenile, this is a not critical period. Why? Because the larvae is not build the structures. Only the structures are differentiation, the functional activities. And this process, it's not linked or relate with the high mortalities. This model present very conservative process. And the question is, where in where stage we can improve the protocols? Only when we are only when we are when the larvae are specialized the functions, not when the larvae are built the structures. This period is conservative. The other period is more easy to reduce the time of development of the larvae. This is a juvenile production of our freshwater shrimp, Griffiops cementarius. This is the picture of the eggs in the last period of the development. This is juveniles, and this video we show you the juveniles that we obtain. This is important to for us, this species, because we work not only for aquaculture, for integrate aquaculture. We work because the fresh water for us um, is critical. We live in the arid zone because for this reason, we need to work with system that reuse the water, like RAS, aquaponic system, bioflux system. And these species are very important because are in a critical situation in the river and are protected, and we can work for repopulation the river with the juveniles. We have a long experience with the repopulation within species, native species for the north of Chile. And we work an aquaculture with the other element at its own. The fresh water is a critical resource for us. The innovative activities in aquaculture involves a small scale and a social and economic and cultural importance of this species. And we work for an aquaculture for the conservation. This aspect is for us and it's very important because we work with the integrated aquaculture with the program that include the fishery and the aquaculture. And we have an important work for the conservation the species in the north of Chile. I don't know. This is our juveniles. Oh, say, we release in the river these juveniles. We mark with the elastomer this juvenile, and we analyze the release in the river, the impact in the river. This is a picture of larvae in the process of metamorphosis for juveniles. This is a migratory species. It's other complex component of the aquaculture. And it's a big work that we have development. And I don't know why I want to invite you, all of us, to work for this aquaculture with different focus. I don't know. This is the finish. I hope that you can understand me. but many aspects to speak. But I am here with my email for the question or um, give you some kind of other information, papers or lectures that we can give you some kind of material for read. So, Elman.
Okay. If you have, as Dr. Maria Cristina said, if you have any questions for the people that are Spanish speakers, para los que hablan español, if you want, you can ask in Spanish. Ustedes pueden preguntar en español. And I can translate your questions. Yo puedo traducir las preguntas for those that are just speaking English, like Cheval from Turkey. I guess that Mr. Burgos has a question. He always has questions.
Hi, Guido. Really, in this moment, I don't have any question. Maybe it's an uncomfortable question. I'm so sorry, but it's maybe it's a mistake with these images, with these pictures, with the species, because Lithopeneus banamei is so different. Because here in Ecuador we have a lot of culture of this type of shrimp, and it's so different than those pictures that you show minutes before. I'm sorry, but maybe it's an uncomfortable question for that. Maybe I have any confusion. It's possible, but because this species that you show is more close to macrobrachium or any similar.
Yes, the Cryptocemeteries is more close to macrobrachium, but I show the Lithopeneus banmei only for the present the kind of catch the juveniles.
Yeah, because we have different species for shrimp for freshwater shrimp, and maybe it's a good guide for to teach different type of technology or or anatomy of the species. But here, for example, here in Ecuador, because we have a four big regions, the insular region, the littoral, the Andean region, and the Amazon region, we have a lot of species. And for that, maybe it's a good opportunity for take any cooperation to work with this poor biology in this type of species, because we need more recognition. For example, we try to work with some students in Macrobrachium brasilensis. This is a species that is distributed here in Ecuador, but they don't have a lot of revenue in the FCR, in the feed conversion rate, and in the growth. And for that is a problem for this cultivation. It's only a comment. Thank you for opportunity, and thank you for remembering me, Dr. German.
No problem at all. Well, we appreciate the speech that Dr. Maria Cristina gave to us today. If you don't have any question right now, her email was in her presentation as well, so you can write to her later on. for any questions. Also, she's open to receive students or researchers to come to Chile and to her lab. And she's doing a outstanding research that I really like it a lot. So this time, I would like just to remind you that we are here in the international SDG network between our three universities, the Universidad Católica del Norte, the Norwegian University of Life Science, and the Bursa Uludağ University from Turkey. And according to our program now, our next speaker will be Dr. Rodrigo Poblete. He will be talking about the recovery of substances of interest from sludge generated in the pretreatment of wastewater from the northern scallop farming industry. And the third speaking will be myself. And our last speaker, he has an injury from an accident and he broke some ribs. So he's not able to do the lecture today. So we will try to figure out when he will be available and we will send you an email for you to see his presentation. So this time, I'm going to leave you with Dr. Rodrigo Poblete.
Hello, good morning. I am Rodrigo Poblete. First of all, I'm sorry for my very bad English, but I'm going to try. Can you see my presentation?
I can, yes.
Okay, thank you. Say the Dr. Merino, I'm going to tell you about the recovery of interesting substance from industrial and real and industrial sludge generated from wastewater treatment of scallop culture or farming industry. As you know, the marine bivalves are very important for the food and also for the economy. In this respect, the north of Chile and the south of Peru also, the culture of Ostión del Norte or scallop are very important. Too many people work in this industry, so it's very important for our economy. And this organic, this scallop is cultured in suspended systems. As you can see in these pictures, the scallop or ostión, we say it in Chile, is cultured in this kind of system. Is a lantern, dried and empty lantern. And here you can see in this picture the lanterns or the pernet in the water. But there is very different this photo from this one. Here you can see the lanterns are full of fouling. Fouling is the accumulation of invertebrates and microorganisms that progressively settle in this system. So the pass of the water and the nutrients for the scallop is very difficult. So it's a problem for the scallop cultures. To solve this problem, the lanterns and the suspended system are taken out from the water to be cleaned in high pressure machine. So use water to clean this kind of system. This used a very high amount of water to clean this system, producing a high amount also of wet and saline sludge. In this picture, to the right, you can see workers that they are working with the high pressure machine to clean these very dirty lanterns. The idea is to clean it to get something like that, very different. In this process is produced, as I said, wastewater and also this kind of sludge, very dark, very concentrated. But here in Chile, also in other parts of the world, we suffer with a drought, the reduction of the rain. It is very important to can recovery water and other systems from this kind of sludge. Here you can see the wastewater on the sludge. There's a problem because the company needs to manage this kind of wastewater and sludge. But as was said, we are suffering a drought that affects the north of Chile and several other places. But obviously, it's very important to produce strategies to recover water from the sludge and other interesting species. One of the technology to produce this kind of recovery is the use of reverse osmosis system. It's a very strong technology, very effective, very expensive too. Also, it requires a lot of energy and excessive maintenance. So we think that it's important to develop other kind of technology to get a recovery of water. An interesting alternative is the use of the thermal process as the multi-effect distillation, multi-stage distillation, and humidification and dehumidification process. Those kind of technology use thermal system to remove the water from the wastewater or from the sludge, and obtain a clean water in other places. In the case of the humidification and the humidification process, we have a hot zone where it produces the humidity, and this vapor is conducted to a cold zone to obtain a condensation system. If you have any question, you can interrupt in any moment. You can feel free, please. This system, HDH system or the humidification system, is a low cost, simplicity. You don't need a very high temperature to produce vapor and can be used in any places. And the other interesting issues is that you can use solar energy to produce the humidification process or the hot zone of the process. But what about of the water quality? As I was saying before, with this system you can get a fresh water, but we don't know if this fresh water is really clean and we can use it to drink or to irrigation process. So the water quality is very important in this case. We need to think that we are working with a real industrial sludge that can be very polluted. So maybe the fresh water obtained in the condensation system could not be very clean. Aspect to consider is the presence of salts, suspended solid, this solid, total dissolved solid, chemical oxygen demand, biological oxygen demand, between several others. Obviously, we are obtaining a distilled water. In this system, we are distilling water. So we can infer that the quality is very good. Really, it is very good. We don't have the suspended solid, the COD, BOD, or TOC could be very low. Also the salts or electrical conductivity. Very good, no problem. But what about with the microorganism? This could be a problem. Again, if you see that, we are obtaining a vapor and also small drops that can transport microorganisms. This kind of transport or microorganism is called airborne microorganism. So we can obtain in the condensed water clean water, but could be the presence of some microorganisms as bacteria, fungus, viruses, or others that need to be controlled to avoid problems related with the quality of this water. It was observed in several studies that in these drops we can obtain some pathogens that can produce meningitis, hepatitis, and several respiratory disorders. The idea is that we need to mature this kind of water. If we detect the presence of microorganisms, we need to conduct a disinfection process. I'm going to present you the case of study that we described in the university here in Coquimbo. We are very near of several scallop cultures. In this case, in Coquimbo, that is around 1 hour to near here. In Coquimbo, they culture Agrobacterium purpuratus, that is a scallop, the ostión del norte, as we say in Chile, that is cultured in lanterns or pernets. Here in this photo, you can see the pernets full of fouling. We have take from the water to clean in high pressure machine to go from this to this. Yeah? But it produces a wastewater and a sludge that we have studied, dried, and recover water from this sludge. For this study, we worked with a sample of 200 kilos and 50 kilograms. It was a very salinity and wet sludge with a 73% percentage of water, and was submitted to a solar drying process. For this, we placed the sludge here in this solar distillation process that has a sunroof walls made from polycarbonate, a transparent polycarbonate, what that has a thermal conductivity of 0.2 watts meter Celsius, and thermal, an optical transmittance of 80%. In brief, it is a transparent system that allows the entrance of solar radiation to produce a greenhouse effect. So the heat produced in this zone is mainly maintained in this area, promoting the production of vapor in the hot zone of the humidification process, and the vapor is extracted using this extractor to conduct to a heat exchanger system and obtain a condensation in a cold zone of the dehumidification process. To promote an air circulation, we use a solar air heater that gets air from the atmosphere and is heated in the system, as we can see in this diagram. In this system, the air is heating and come inside of the solar drain with higher temperature. To compare this system another, we use a parameter that is called amount of accumulated solar radiation that allow us to quantify the amount of energy that is used in the system. For this, we get a series that use the quant of energy in time n minus 1 plus the time of measured multiplied by solar radiation, the area of the system, and the mass of the sludge. So we have a series of time, and we are quantifying the solar radiation accumulated in the system. The coefficient of performance of the solar air heater is determined using this formula that is a ratio of the energy of the solar air heater divided with the power of the blower used in the system. The amount of energy used by the solar air heater is considered the velocity of air, the density of the air, the area of the solar air heater, the heat capacity of the air, and the difference of temperature between the outside and inside of the system. So we can consider it the heat system as a quantity of energy where we are going to coming into inside of the solar dryer. Any question at the moment? Okay, as was said before, it's very possible that in this water we can obtain some microorganisms that we need to control and disinfect. For this issue, we compare three kind of system in three reactors. One of these use a UV lamp. The other one use a UVC lamp, and the other one use a UVA or UVC lamp plus ozone. So we have this system that we can compare the efficiency of the disinfection process. UVA lamp use a lamp that emits a wavelength of 365 nanometers of UV. For UVC, we use a lamp that's emit to 254 nanometers of UV wavelength. As you know, it's very important to consider the amount of energy used in this process and also the efficiency of this usage. So the performance energy is very important considering the cost of the process. For this aspect, we use a parameter that is called electrical energy per order. It was a formula created by Bolton et al. That consider the power of the equipment, the time of use, and also the volume of the water to disinfect, and the concentration in the beginning and in the final of the disinfection process. So we expect to get electrical energy per order very low. If it is low, it means that we have a low energy consumption and very high disinfection process. The results of this case of study show us that very high temperature inside of the solar dryer and also in the solar air heater. In this plot, you can see the temperature of the air inside of the solar heater and inside of the solar dryer. Obviously, the peak is related with the middle of the day in a sunny day, and the low values is related with the behavior in night, or also in a day with a low solar irradiation. Considering the coefficient of performance of the solar heater, we get a very interesting value of 3.9. It is very good because say us to we get a very high temperature difference and very low energy consumption. This is a very good news. In the bottom plot, we can see the temperature of the ambient inside of the solar diode and the solar irradiation. What about the water recovery in this system? It was very, very good. We obtained a very high amount of water. We need to think that in this system, we get 3 kilograms of sludge in the system, going from this value to a final value of around 1.5 kilograms of sludge. So we get a very good dryer process and also a water recovery from that. But we measure and control the volatile compounds and the airborne microorganisms obtained in this process. We obtain these values of bacteria concentration, CFU per millimeters. So we need to disinfect this process. And you can see the evolution of the concentration of bacteria in the time after 1 hour of disinfection process. As you can see, the best performance was obtained when we used UVC and ozone system. We get a very good performance, very fast too. So it was a very good result. To understand this very good performance, we need to consider that also of the effect of UVC and ozone, we are producing hydrogen peroxide. As you know, this reagent is a very good disinfect products that allow us to get a very good disinfection system. So inside of the reactor, we have UV, we have ozone, and also the production of hydrogen peroxide. But we inferred that. We hypothesized this production, and to confirm this behavior, we conduct an experiment using clean water and measure the production of hydrogen peroxide under this condition of ozone and UVC. And in this plot, you can see the production of hydrogen peroxide and also the temperature of the system. What is the reason of this heating system? What also the UVC lamp emits heat, and this heat is dissipated in water and is related with the production of the ozone, and also is reported a very good diffusion of ozone in the water, enhancing the production of hydrogen peroxide. Also, the generation of hydrogen peroxide is also related with the formation of hydroxyl radical. The hydroxyl radical is the base, is the focus of the advanced oxidation process. In fact, it's very difficult and expensive to measure hydroxyl radical. It's very expensive to measure it. It's very complex to measure it. But indirectly, we can infer the production of hydroxyl radical across the measure of hydrogen peroxide. Why? Do this equation. In presence of hydrogen peroxide, we can obtain hydroxyl radical. And as you know, hydrogen peroxide is a very good disinfect product that reacts with the cells that can destroy it. What about the water quality of the distilled water? At the beginning, from that, we take samples and we can measure that it has microorganisms and also some a little of organic matter. As you know, as you can see in the bottom plot, we have an initial concentration of total organic carbon that is now zero. Yeah? Why? Because is it possible that some volatile compounds that travel with the vapor and condensed in the distillation system. So we have a microorganism and also some organic matter. Along of the disinfection process, we can obtain a reduction of the bacteria, but also we observe an increase in the concentration of total organic carbon and chemical oxygen demand. Why is the possible reason? We think that in the beginning of the process, and along the bacteria destruction, some organelles inside of the cell is exposed to the water. So maybe we in the water at the beginning we have proteins, DNA, enzymes, and other cellular components that increase the concentration of organic compounds in the water. But after that, in the minute 40 and 60, we observe a reduction of that. And this reduction also is very fast when used UVC and ozone. This is a very good reason, a very good news, sorry. As we said, the generation of ozone and UVC produce hydrogen peroxide. This hydrogen peroxide, in presence of ozone and UVC, produce hydroxyl radical and oxygen. That has a very good performance of disinfection that allow us to obtain a very good water quality. What about the energy performance? That it was calculated using this formula. Here you can see the plot of the figure of merit, what was calculated using this formula. As you can see that UVC and UVA has the more efficient energy consumed. That is related with the power of the equipment and also the disinfection performance. And when we use it ozone, the efficiency performance is not very good, but very efficiently in respect to the disinfection process. And what about the sludge generated? This sludge has these measures that we can observe that has a very high amount of nitrogen, phosphorus, potassium, boron, also has amount of copper and iron. But could be used for application process, but we need to consider the presence of these metals that we need to be removed. Final considerations, the water in the sludge after the cleaning of the long times can be recovered in a humidification process. And we need to be disinfected. The solar steel was improved using the solar heater that allow us to increase the temperature inside of the system. We get a very good amount of water recovered that allow us a very good solution in a critical region during the scarcity of water. We get a very high disinfection efficiency using UVC and ozone. We need around 60 minutes to this process. The second efficiently process was the use of just UVC. The ozone allow us an enhance in the disinfection process, but also a high energy consumption. And this is, it could be possible application of the dried, sorry, it's dried, not dyed, sludge in the agriculture issues. This is my presentation. If you have any question, please tell me.
Thank you, Rodrigo. There are some people also from Ecuador. So you can also, if you want to speak Spanish to them, if the people from Ecuador cannot speak any English, we translate the question for the people that are listening from Turkey. Well, it seems, parece que es una mañana muy callada en Ecuador. It seems to be a very quiet morning today for everyone.
Maybe it's too earlier in Ecuador.
Yeah, I can read the messages, but there are no questions in the messages, Rodrigo. There are
Maybe maybe
from,
every Burgos has a question.
as always.
Yes, I am thinking a question. Yes, it is true. That what's happening with a freshwater ecosystem with fouling? Because
please.
I don't know anything about fouling in freshwater ecosystem. Don't
The
exist.
fresh water obtained in the distillation process.
Yeah.
Yeah, the quality was very good. I show you some results, but this is just a resume. Where is here? Here we have a low concentration of total organic carbon. It's very low, but is so. We didn't get, we didn't observe total suspended solid. It was expected, so it's very good. But yes, we have some total organic carbon, is organic matter. So we need to, well, we can use this water. The problem is not the organic carbon. It's very low. The real problem is the presence of a microorganism. This is a parameter that we need to control. This was the reason of the disinfection process.
Yeah, well, because when you have I will speak in Spanish. Excuse me for
No
the people
problem.
that speak English, but
It's,
it's
it's
better
better
now for
for
this
me.
type of question. Yeah. Eh, en, en jaulas flotantes, nosotros en ecosistemas amazónicos especialmente, no se tiene como tal fouling, pero se tiene un tipo de microfilm bacteriano, digámoslo así, que induce a problemas tal vez en producción. El problema es que nuestros ciclos de producción en peces tropicales especialmente son mucho más cortitos. Estamos hablando de engordes de 6 meses, de máximo un año. Entonces no se evidencia porque después del engorde lavamos las estructuras cuando toca ser lavadas y otra vez empiezan a trabajar. Y esto lo he visto, digamos, en Perú, en Colombia y especialmente en Brasil, donde hay ese tipo de cultivo. En Ecuador hay muy poco, lo hemos hecho experimentalmente. Entonces el tema es cómo lograr, digamos, hacerlo más que todo en mar, porque usted está trabajando en mar y nosotros también tenemos linternas en mar, pero al menos donde yo trabajo el tema es casi todo orientado a agua dulce.
Mm,
Es claro, como usted dice, si hay bajos nutrientes, va a haber poquita capacidad de fouling o de
sí,
sludge, digámoslo así.
sí, sí,
Ahora la pregunta crucial es cómo aprovechar el sludge como sistema multitrófico.
sí, sí. Bueno, lo primero, decir que, claro, en
Before
nuestras
that,
aguas, sí, sí.
just the question is you can do it in English
Thank you so
for
much.
the
Sorry.
yeah. Rodrigo, can you redo the question in English, please?
Uh, I am not very sure, but I think the question that R. Burgos, sorry, what is your name,
Ricardo,
Ricardo?
you, you,
The
you was
question
my,
of,
my, my as well was my teacher in maybe in first or second year of the doctorate. I am now presenting my thesis degree.
okay,
Maybe
nice to
you
meet
don't
you.
remember me, don't problem.
Okay, the question of Ricardo is how can use the sludge? Can we uh, uh, take provecho? No sé cómo decir provecho. We can use the fouling. This is
Mhm.
okay. As I show you, the 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 dried fouling uh, is very interesting. So it has a lot of compounds that we can use maybe in agriculture system as a fertilizer. Fertilizer. I am not a specialist in fertilizer. I think that Herman can give you some ideas, but I think that the presence of nitrogen, phosphorus, potassium, sodium, magnesium, calcium is very high. So maybe if we control copper and iron, could be used as a fertilizer in agricultural application, I think.
Thank you. Thank you so much. Maybe in other words, thinking in English,
Hmm.
the other question is how can use this sludge or fouling like a food in multi-trophic system in aquaculture?
The food, I am not a specialist in that. I think it is as a aspect that we need to consider it. Maybe in a next project we can abort this, but could be very interesting, really.
Thank you. Thank you a lot because this is very sincerely and maybe we can use this type of uh, material as other source of food or the nutrients recycling in the aquaculture system or others similar.
Yes,
Thank
of
you so
course.
much. A
Thank
good
you.
presentation.
Thank you. I tried.
Well, thank you very much, Dr. Rodrigo Poblete, for your presentation.
Thank you.
So that was our second presentation for today, and we'll be continuing with the next one and the last one for this morning. Just let me a few minutes. Okay, so I think I am ready for everyone. So is everyone able to see the slide? Dr. Burgos, can you see my slide? Yes? Or is anyone listening to me? Maybe I lost connection.
We see the slides.
Okay, thank you very much. Well, my presentation will be regarding biosolids, mineralization, and biofiltering microbiota in a pilot scale system with rainbow trout, river prawn, and lettuce. This is an aquaponics pilot system that we have deployed in some high schools and also here in our university. This is a work that has been done by me, myself, of course, Elizabeth von Brand, Christopher Concha, Norma Perez, Camilo Merino, all of them from Universidad Católica del Norte, and Doctora Constanza Hanna, Dr. Carlos Cortés, and Engineer Alejandra Duran from INEA, both Chilean institutions. And all this research belong also to our partnership with the University of Life Science from Norway and the Bursa Uludağ University as well from Turkey. Well, this is my agenda for today. I would like to introduce you about the problem and the explore solution that we have done. What is the aquaponic water quality that we have been reading, the experiences with the river stream and the lettuce, river stream that already Doctora Marina Cristina talked to you. Then we are going to talk about some mineralization approaches for the slush that the fish produce in the system. We're also doing some microbiome exploration to have an idea what kind of bacteria we have in our system. We are just starting to get that. And also would like to show you a little thing about the university and the high school that we are working with the aquaponic systems. So basically the problem that we are facing here in the region where I live, Chile is a very long and thin country, as you may know, and we are in the north of Chile, about 500 kilometers north from the capital, Santiago. And as you are able maybe to see here, there is on the left and in the left corner, there is a map of the region where I am. The region is called Coquimbo. And at the very beginning of the year 2000, about half of the region was having or facing a scarcity of fresh water that is colored in red here. But 17 years later, pretty much the whole region is in red, meaning that there is scarcity of water. There have been some studies on the rivers that we have in the north of Chile, and I point out here the river that we have in our region. There are these three ones here. This is the Limari River, the Elqui River, and the Choapa River. And this graph is showing us what is the need for water to produce food or for use the water in other industrial processes. So what means in this case 43% means that we are requiring 43% more water than the river already had. And for the Limari River, we are asking for 80% more water than the river already had, meaning that our technologies that we're using maybe are not efficient in the use of the water, and we need to start looking for other kind of technologies to do an efficient use of this water resource. Because we were facing this scarce of water in the region, we developed some dams, so we were able to catch very fast the water that was melting from the ice in the mountains, and we keep it. So in this picture, you are seeing that this dam was pretty much full, and because it was too full, some of the water was spilled out from the dam. But at the next year or in a few years, the water was looking like this. So we didn't have too much snow in the mountain, so the melting of that very small amount of snow was not enough to fill up the dam. So by year 2012, the dam was working at his 25% capacity. But the thing became even worse because by the next year, the dam was looking like this, pretty empty, a very small amount of fresh water available. Today, we have a little bit more water, but still, we're facing this situation with water scarcity in our region. In this graph, you are able to see about 100 years of recording regarding the annual precipitation in our region. So at the beginning of the 1900s, we were having about 170 millimeters of precipitation. But as you see, along all the years, this line of the slope was negative, and we were even having even less precipitation, water from rain. And at the beginning of the year 2000, we were having about 80 millimeters of annual precipitation. And today we're around the 50, so even less than at the beginning. So every year we have even less water. And in the chart that I have here on the left and the right, actually, I want to show you how much water do you need when you want to produce 300 grams of fillet. Let's say you want some 300 grams of a hamburger that is delicious, maybe, but you want that, you might need about 4,500 liters to produce 300 grams of fillet. But if you would like to have 300 grams of fillet from a pork, then you will need about 1,400 or so of water, liter of water. And if you like to have a chicken, 300 grams of a fillet of chicken, that will be roughly 11 and 7,000 of liters of water. So as you can see there, depending on what kind of protein you are producing, you might be requiring more water or less water. So, and here comes what we are working right now, because this region is also an agricultural region. The the agriculture engineers have been working, studying, and developing hydroponic systems. And I can show you the picture right here, basically with lettuce. And we, as a university, we have been working with something that we call it aquaponics. Maybe you are familiar with that. And the aquaponics, we actually are using the same water to grow lettuce and also to farm, in this case, rainbow trout. And of course, we are also including this little friend here. This is the river prawn that Dr. Maria Cristina Moral just spoke to us a few minutes ago. So to do that research in aquaponics, we set up different systems in different places. First of all, we started in our university. So we deploy 4 different aquaponic systems, actually three aquaponic systems, and the fourth was just a recirculation aquaculture system, just to compare how was performing the recirculation aquaculture system in comparison with an aquaponic systems. And in the INEA, that was the other researcher institution, we also deploy over there 2 aquaponic systems to do our research. We also deploy similar system in 5 high technical schools that I want to show you later in this presentation. So the idea that we have behind was to use a rainbow trout as our target fish. And that is very interesting because in this partnership that we have with Turkey and Norway, both countries, they also have rainbow trout, so we can share our knowledge with them without any problem. If we were using other fish, maybe it would be not that easy to share the knowledge. But basically that we have here when we're doing aquaculture, the fish will excrete ammonia and also they will produce biosolids. So the ammonia in this system will go through the biofilter. These 2 towers here are working as a biofilter. One of them is working as a trickling biofilter, and the second one is working as a submerged biofilter. And the biosolids are captured in this settling device, and we keep the solids in this settling device for a few days. So to happen here a mineralization of the solid, and maybe with that mineralization, we are looking the productions of some nutrients and macronutrients that will be like this. So if we have these macronutrients, nitrogen, phosphorus, potassium, calcium, sulfide, and magnesium, those are very important for the veggies, in this case lettuces. And also we have micronutrients, iron, chlorine, manganese, boron, zinc, copper, and molybdenum. That will be very nice because those nutrients will be used for the veggies, and they will be taking out those nutrients and cleaning the water, and the water can be recycled back to the fish again. That is the model that we were looking for. And also we were exploring the river prawn to put those animals in this system, and we decided to put these river prawns with the lettuce in the same tanks. So we move forward on this idea, and with the other students that we have in our team that are more focused on instrumentation and control, they starting to build a system based on IoT. Sebastian Messina is already sitting and seeing the presentation. So if you have any question at the very end, and he's still here, maybe he can answer some of them. But Sebastian and Maximiliano, they were very interested in to design and build a system that can monitor some of the variables that we were looking could be interesting for the aquaponics. Some of those variables were atmospheric variables, and others were water quality variables. So in terms of the water quality variables, we were looking for the water temperature, for the water oxygen saturation, and also for the water pH. Of course, could be very nice also to have another sensor that would be able to record the conductivity, but we didn't have that in our system. And also we were recording for the atmospheric, the ambient temperature, the ambient humidity, and the light intensity that we're measuring luxes. So basically, that was allowing to us to follow some of the variables on the system. Well, the first thing that we were doing here was trying to understand what happened with the water quality along the year. And here, this graph are showing several days for this. We're starting in October 2023 and finish the recording on March 2024. As you can see, the pH in these systems for the 3 aquaponics system and also for the recirculation water system, the pH was pretty stable around 8. The alkalinity was going up and down. That is very common in the recirculation system because the activity of the nitrification, meaning that we need to add some chemical to control the alkalinity. The temperature was pretty much stable. We would say in all these systems we have heat pumps, and we set up the heat pumps at the very beginning at 15 degrees Celsius, and then we increase it up to around 20 degrees Celsius. So you can see that those changes were following the temperature that we set up. Here we have a problem in one of our technical school that during the summertime, the water temperature, as you can see here, was close to 27 degrees Celsius. And what that happened, all the rainbow trout actually died. So temperature is a key factor when you're doing aquaponics or with rainbow trout. See, if temperature is too high, maybe you would like to change to other kind of species that are able for higher temperatures. But other water quality condition that we were following was the ammonia. And the ammonia was most of the time very low. We have some spike here for some reason, but mainly low because the biofilter were working nicely good. The nitrite was also low. We also have a little spike here for some reason. We were still wondering what happened. And the nitrate was, as we were expecting, was increasing along the time. That was pretty good because this will be the nutrients that the veggie will be using later. Also, we saw that the phosphate was also increasing in the water quality in the system. Sulfate is naturally present in our water, so it's nothing that the fish are really producing in there. Magnesium was also showing in some of the systems. So these are how they look like the lettuce production in one of the high schools, and these lettuce were coupled with the production of the rainbow trout. So you can see nutrients are in the water. We're also looking for calcium and potassium. Basically, calcium was pretty much absent most of the time. The same thing for potassium most of the time. And this spike that we have here and the spike that we are also showing here is because we started to add calcium and add potassium when we were working with the alkalinity. So when we need to increase the alkalinity, we use calcium hydroxide or potassium hydroxide as a source for alkalinity, and also as a source to introduce the calcium and the potassium for the system. And here are the juveniles of the river stream that we were putting along with the veggies in our bed system. Here are the boxes and the bag with water and the shrimp that we were putting with the lettuce in there. How was performing our rainbow trout? This is our system that we were just putting together. Very old pictures right now. The fish that we buy from a nearby hatchery, those fish, the rainbow trout here are about 20 grams each one. And here we can see how was the growth rate, or during the time it took about 10 months for all the fish, actually between 8 months and 10 months, for all the fish to reach 500 grams. That was our target weight. Very nice and very interesting that the fish were growing and the plants were growing and the nutrients were present in the water. So these are pictures that we got from our system at the university. You see the lettuce over there, the little basil that we were working with. And here are the river shrimp that Dr. Maria Cristina was producing in her lab. Those are 3 months juveniles, and we put those juveniles in our aquaponic systems for them to reach a higher size. So they were 3 months. They were growing in our system for 5 months, and they got this size. We moved them into the veggies bed. So we put in the shrimp over there, and on the top we put the veggies that you can see on the bottom over there. And later on, we were harvesting the shrimp that were growing with the veggies. So interesting scene over there. The question that we have on that time was how many shrimp can we put together with the systems where our lettuce? Because the shrimps are omnivorous, meaning that they can eat meat, but they also are able to eat veggies. So we did an experiment with different stocking densities. Some of the tanks were having 10 shrimp per square meter, other 5 shrimp for a square meter, other 14 shrimp for a square meter, and some of them 17 shrimp for a square meter. And we were following during several days how was the length of the root of the lettuces that we have in the tanks to figure out how was the lettuce performing together with the shrimp, or excuse me, with the prawns. And we saw that at the smallest stocking density, the lettuces were growing much better than the higher stocking densities of prawns. So all these graphs are showing that when we have between 5 to 7 prawns in the tanks, that was no problem for them to be growing in the same system where the lettuce were also growing. But if we have higher prawn densities, then the veggies will be pretty much without any roots. So here you can see some of the veggies that were losing pretty much all the roots, but over here you see more root developments in the other cases. So that was very interesting to start, and we are planning to continue with this research this year. And regarding the micronutrients, it was very difficult to find micronutrients that were in the system itself. So we were looking for zinc, molybdenum, copper in the recirculation system, and also we were looking for the same micronutrients in the 3 aquaponic system that we have deployed. And as you see here, the numbers or the concentration are very, very slow, very, very low for each nutrient. A little bit of increment on zinc, but not much on molybdenum or copper. That is what we learned on what was going on in the water quality in our systems. In terms of nitrite, here says cycle 1, cycle 2, cycle 3, cycle 4. That means cycle of production of lettuce. Lettuce is take about 4 months, or excuse me, 4 weeks or 6 weeks to reach the commercial size. So these are 4 cycles of production. And as we move on, we see that basically in all the system, but in the system, in one of the aquaponics system, the nitrate was highly, but it's not a problematic for the features stream to have 350 milligrams per liter, but basically was reaching about 100 milligrams per liter in all systems. That is very healthy for the lettuces. Calcium was also in a nice amount of quantity required for the veggies. So that is the interesting thing that you were able to see in the picture, that the veggies were growing very good. The only thing that it was it's not too good is that in all our system we have a pH that it was pretty much close to 8, and veggies are very efficient into uptake nutrients when the water pH is between 5.5 and 6.5. But we were over 6.5. So maybe we should look for some way to reduce the the pH in our system, so the plant will be more efficient into uptake the nutrients, or maybe have an aquaponic system that people today call decouple, meaning that the aquaculture system is working isolated from the agriculture side, and the only connection will be for a few hours during the day when water with nutrient is moving from the aquaculture system to the hydroponic system. But in our case, at this time, we are working with a couple system, meaning that the water is moving from the fish to the plant, from the plant to the fish in an hour or so, several times a day. Well, this is the context when we see an aquaponic system. We have basically our fish that we are growing. Those fish will produce some nutrients that are dissolved, and also they will produce some nutrients that are not dissolved. Those nutrients are within the solids that the fish produce. So what we have seen that other colleagues in other parts of the world, they are taking those solids and they work with the solids in a separate bioreactor. So in that bioreactor, they will produce the nutrients, and then those nutrients will be put back into the system. So the system will be then use those nutrients and also the ammonia that have been converted into nitrite in the nitrification system. And with all those nutrients, the veggies that are growing in the hydroponics system will be uptaking them. And of course, because they do that, the water will be in a good quality to go back to the fish. So basically, what we have done then is to take the solids and to figure out if we can do mineralization aside from the system. These pictures do not belong to us, but it's just to show you what basically you will do if you have the fish tanks. Then maybe you have some technology, let's say a drum filter that will remove the solids. The solid could be collected in a tank. Basically, if you want to remove some of the water, you can press those solids so that you can have some kind of slush that is with less water. That is basically what the most companies will do here in Chile, growing fish. In our case, we were doing from the collection tank, we would take some of the sludge and we were performing some studies to determine if what happens if we do the mineralization in an aerobic system or if we do it in an anaerobic system. This is a small video when we were just starting with the recirculation system. In this case, we didn't have any aquaponic, but we were having the two nitrification towers. And here is our radial settle. And all the solid that we capture, we move it into this bioreactor where we keep it for 24 hours with aeration. And after that, we stop the aeration so whatever solid that we have in there settle down, and we just remove the water with the nutrient that we have in here. And then those nutrients, we put it into the aquaponics system. Basically, that was our first attempt for the bioreactors. But with that attempt, we started to do some research in small vessels. Those vessels or the bottles that we have here, they were about 5 liters, excuse me. And we did an aerobic treatment and a facultative treatment, meaning that in this case, in the aerobic treatment, we put an airstone in each of the bottles. And we were looking at the nutrients on time. And in the facultative treatment, basically that we did is we removed the airstones. And we keep some device to keep the liquid that it was within the bottles in movement, but without any oxygen addition, to see what happens in those conditions with the nutrient production. So the question that we have at the beginning of this experiment was, is what will happen with aeration or without aeration? Which will be the best treatment to be able to remove the nutrients that are within the solids? The second question that we have is, if we are able to do an incubation of this sludge in a sequential bioreactor that could be starting aerobic first and then anaerobic. Then we were looking if there was any effect on the mineralization process if we were having different sludge concentration. Let's say we started with a 20 milliliters of the sludge per liter, 30 milliliters of sludge per liter, 50, 16, and 80 milliliters of sludge per liter. We didn't did a higher concentration because back then we didn't have a strong blower to provide us enough aeration to keep the sludge moving in the water column. So we just finished up to the 80 milliliters of sludge per liter. And the last question is the one that it should be for. We were also wondering, as I said before, for lettuce or for the veggies, the best pH for them to be able to uptake nutrients between 5.5 and 6.5. So we were wondering if there was any effect and for the nutrient release from the sludge if we do the incubations of the sludge at pH near 6 or near 7. And we were measuring the ammonia, nitrite, and from the macro nutrients, we were looking at the nitrogen and phosphorus, sulfite, calcium, and magnesium. And from the micronutrients, we were following iron, copper, and zinc. And we were looking also at pH and ORP, the oxidation reduction potential of the water. So these are our first results. And we were doing 3 replicates per incubations. These are bioreactors that are 2 liters. In the left side, we have here the treatment that was aerobic, meaning with the earthstones within them. And on the right, we see the anaerobic treatment without the aerations in the bottles. And basically, we see that iron and zinc, both of those nutrients were not increasing in concentration, whether they were in aerobic or whether they were in anaerobic conditions. But in other cases, magnesium, it was disappearing if we have in aerobic, but was increasing when it was in an anaerobic condition. That's an interesting thing. Same thing for calcium. Calcium, even that the fish do not produce calcium, and the calcium was provided along with the alkalinity, we see that in aerobic condition, the calcium disappears, and in the anaerobic tend to appear a little bit. Phosphate, we see that it was more important, the presence of the phosphate when we were doing anaerobic in comparison with aerobic, just because the release of the phosphate requires 2 phases. First, it had to be into aerobic conditions, and then it had to be into anaerobic to be released into the water. So that was, we were wondering if we can have maybe 2 bioreactors and do the sequencing aerobic, anaerobic treatment, because we knew that the release of phosphate will be after the process of an aerobic conditions. And about ammonia, well, the ammonia was increasing in aerobic conditions. And then we start looking what happened with the sludge if we were working at 20 milliliters of sludge per liter. And here, each of the graph will be having half of the graph will be doing an aerobic condition within the first 150 hours of incubation. And later or after that 150 hours, we continue the incubation of the sludge, but in an anaerobic condition. Remember that the anaerobic for us was that we remove or take out the earthstones. So any aeration or oxygen that was in the system was depleting along with the mineralization process. But we can see here when magnesium was increasing during the aerobic phase, but it started to decrease within the anaerobic with a little increase as well. Calcium was pretty much the same. Phosphate, we were saying, was a production in anaerobic condition. Nitrate, was in aerobic was disappearing, but during the anaerobic process was disappearing, and probably because some denitrification that was growing over there. Potassium was more present during anaerobic. Ammonia was very low. pH was, as I said, it started with 7.5, then increased it to 8, but during the anaerobic process started to decrease a little bit. Our RP was always over 0 when we were in anaerobic, but when we turned it into anaerobic, the RRP was reducing below minus 100 millivolts. This is important because in our natural water, we have sulfate. And the sulfate, when the RRP is below 150, about that much, the sulfate can be converted into hydrogen sulfide. And that hydrogen sulfide is very, very toxic for the fish. So if you do mineralization in an anaerobic condition, you need to look which are your RRP measurements so to avoid this sulfide to be converted into hydrogen sulfide. And this chart here, we have numbers that you can be following what happened, for instance, for any of the nutrients that we were looking at before we start the process and during the aerobic incubation, and also what happened during the anaerobic incubation. In some circumstances, you'll see that the nutrients increase or decrease, and others just maintain without any changes. But what we saw basically with the replicates, that was very difficult to have a very steady results. The replicates were very, we have a range that was a wide range of results. Also meaning high deviation as well. So more research is needed to be sure what is going on in this situation. Well, if we increase now the sludge from 20 to 30 millimeters per liter, results are basically similar to what we saw, but of course we have more nutrients in comparison with what we're having with the 20 liters. So that is what basically that we were having here. Well, I'm going to speed up a little bit because we are reaching our time for our seminar today. And here we have an experiment where we were doing the sludge process with having a concentration between 60 and 80. So in this case, the 60 will be the green line here, and the 80 will be the blue lines. And if you follow all the graph, you'll see that you will have more nutrients production in pretty much in all the experiment that we had when we were doing the 80 millimeters of sludge per liter. So that is interesting. But as I said, we didn't have a powerful blower to keep up all the sludge in the water column, so we couldn't continue on experimenting with higher stocking of the sludge. But the trend in here that we got is the more sludge you have, the more nutrients you are able to produce, and all the nutrients can be later moving into the aquaponics to provide the nutrient that the veggie will require. And in this time, we're also looking at the RRP, but as you see, the RRP never went below 0. So that was good, because meaning that the sulfate became as a sulfate, didn't convert into hydrogen sulfide. We also did, as I said before, an experiment regarding the effect of pH in mineralization. We have a pH about 6 with the blue lines and about 7 with the red lines. Basically, for some nutrients, it was very nice when you were working in acidic environment. But for potassium, you can see here, it was much better when you were close to a neutral pH. So what is showing this to us that the pH will be important when you do the mineralization. A sludge concentration and pH would be key factor when you are building or designing the bioreactor to do the mineralization of the sludge produced by the fish, and then you get nutrients that you can use in your hydroponic or your aquaponic system. So at the very end, now when we can see what happened with the aeration and no aeration system, well, we were able to see that the anaerobic was much better to produce nutrients, but because it was anaerobic, we were having small odors around, so something to take care of. It could be interesting to do a sequential bioreactor system, meaning aerobic and anaerobic later. Yes, we see that it's interesting to do so. And you have to have first the bacteria for those environments available so you can start very fast your bioreactors and the transformation of the nutrients. Do we see any effect of the large concentration on the nutrient production? Yes. This we saw that. And also we saw that the pH have an effect. It will be better to do the bioreactor mineralization in an environment with more acidic conditions. Well, let's move on here to our technical school. We deploy, as I said before, the aquaponics systems, all of them the same technology. We deploy in 5 different high schools. The 2 research institutions were providing the trout or the river stream or prawn to any of these high schools, and also with the other institution, INEA, they were providing and advising about the hydroponic management. So this is basically the system. It has 2 tanks, the 1 cubic meter tank each one for the rainbow trout farming. It has 4 bed systems with for the hydroponic productions. Here you see the picture as it look like. And this small unit here is having the 2 biofilters, the 2 towers. Here you can see them. The settling device to remove the solid that the fish produce. The sump where the water will be returning from the bed where the veggies are growing. And in this sump, we have a pump that take the water and move the water to the biofilters and from the biofilter back to the sump. And the other part of the water is going to the fish, from the fish to the settle, from the settle tank to the plant, and from the plant going back again to the sump. This unit you can put more fish tanks if you want. And also we created a handbook of these modules for the high school to read and to learn how to operate this system. This is our region. You see here where they were deployed along them. We were here, La Serena, Coquimbo. Some were in Ovalle, some of them in Punitaqui, and the southern one was located in Canela. In there, we were looking now how the lettuces were growing in those areas. We put about 30 lettuce per square meter, meaning that for each bed, we were able to put 90 veggies. Each bed had 1 meter width and 3 meters length, meaning that we have 3 square meters per bed. We were looking at the growth of the leaves and also the growth of the roots in those systems. We were also measuring the foliar green cover. This means when we put the little plants, they look like this. But when the plants are growing, you see that they expand the leaf, and when they cover the whole bed, it will be the time to do the harvesting. The result that we had here were dependent on the location. This was January 6, January 12, January 19, and January 23. 1 month of growing lettuce. Same dates over here, two different places. Tongoy, that was in the middle zone of our region. Pan de Azúcar, also the middle zone of our region, but it's more inland, and Tongoy is close to the coast. And we see here that we got a better lettuce growth when we're in the system that was close to the coast, maybe because the temperature in the environment was colder in comparison when we move into inland, when the temperature is too high and maybe not good for the lettuces. Also, we were looking then, how was the growing of the lettuces depending on which hole they were seed. This is we're looking here at the bed from the top. Here, these two here are 1 square meter, here the 2 square meter, and here the 3 square meters. So we put plants on each of these holes. We call it, this is column 1, columns 2, 3, 4, and 5. The water goes in right here and then flow all the way to the end, and the water goes out from this side. So we were looking then how the hydraulic of that bed system will be maybe affecting the growth of the lettuces. That means some kind of engineering. If you like engineering, you can also maybe start continuing doing this research. And we saw this when we were about in January 4, the plants were measuring about 45 millimeters of length, but some of the plants were larger, the ones that were in the column 5. For some reason here, all the plants that were here were the larger ones. The smallest ones were the ones that were in the column 3, means in the middle. That was happening. And a few weeks later, the trend was continuing the same. The larger was still the plant that were growing in the number 5, in that column, and the smaller one were in the middle, meaning that something was going on in the systems in terms of engineering or in terms of hydraulic. Similar thing. So we ran a research here to do a model, how was the water distribution. And in here you see that the entrance of water and the red colors and the yellow colors and green colors, meaning that we have an important water velocity here. But the light blue or the blue are meaning that the water, especially the blue, means that the water is moving very slow in there, and the light blue is meaning that the water is moving fast. So somehow we had different water velocities, and because that water velocities, we were having these different growths in the lettuce. Here you see the lettuce that were in the middle are growing slower than the lettuce that are growing in the lateral sides of the tank. So if you are interested in this, it would be nice if you can help us to improve the design and the hydraulic or the water movement in the bed where we are doing hydraulic, where we are farming the lettuces. In general, we have better performance in Punitaki, and that's the college, in comparison with the other places where we were doing the aquaponics system. That it doesn't mean that the other places will be changing in anything to reach the performance that Punitaki has. It's just a geographical location. In other words, we shouldn't expect that the aquaponics will be performing exactly in the same way in different geographic places. So this study is showing us that geographic place or location will be also influencing the production of our aquaponics system. So this is our aquaponics and the hydroponic that we have in the institution that we call INEA. Just a general picture for you to see. I guess this will be our final reviewing here. In our university, we have 3 aquaponics systems, but in this experiment, we were following 2 of the aquaponics systems. And also we have a 1 recirculation system. So in our aquaponic system, we have these raceways with a division in the middle that allows the water circulation, kind of a circulation water. And in this tank, we have the rainbow trout. Then we have the same training unit that I showed you before for the high technical school. We have a sump, a radial settle, and the biofilters. And we have two bed tables for the hydroponic growing of lettuce that are growing coupled with the fish tanks. For the recirculation system, the sizes are the same. The only difference with this aquaponics, and in this case, we don't have the bed system for growing hydroponic plants. So this research was done by Dr. Concha. I'm not the expert in here, so I will try to do my best. But basically, we were using a machine that we call Mine Ion. And in that machine, we are able to run some experiment. And with the experiment, we were able to know the species that we have in our systems. And here's the methodology that Dr. Concha is following. He do the extraction from the DNA of the sample that he's taken with the microbes. Then they run that information in some genomics libraries. And with those genomic libraries, he can do the sequencing with the nanopore technology, with the MinION, as I said before. And with that, he's able to get the species that we have in our system. So he was looking what bacteria we have in our biofilter towers. As I said before, we have a biofilter that is working as a submerged biofilter, meaning that the media is always submerged in water. And then we have another biofilter that is connected in series that is working as a trickling biofilter, meaning that the media is not all the time submerged. Basically, is below a raining of water. And that is the trickling biofilter. So in here, A and B are the biofilter for the recirculation aquaculture system. C and D are the biofilter for one of the aquaponic system. And E and F are the biofilter for the second aquaponic system. And this big triangle here is telling us that basically there is very small variability of bacteria in terms, right here, is between 0 and 20, that's the variability of bacteria that we have in the two towers located with the aquaponic system. In the other system, the same thing, very small amount. And in the case of the recirculation aquaculture system, we see that there are more diversity of bacteria in the trickling in comparison with the one that is working as a submerged. That's a very basic information there. And if we move forward to see the variety of bacteria, so in this picture here trying to tell us that these two biofilters are located in one aquaponics, the other two one in the second aquaponics, and these two biofilters with the recirculation aquaculture system alone. And the same thing in basically what we found that in the submerged biofilters, I mean, these are the charts, the most dominant bacteria were those that belong to the class alpha proteobacteria. And the trickling biofilter, the dominant bacteria were those called gamma proteobacteria. And those groups, the alpha proteobacteria and the gamma proteobacteria, are related with the nitrogen cycle. So that's interesting because those biofilters supposed to do the nitrification, and most of the bacteria that we found in there are bacteria related with that process. So basically, that is what we have for now, and we are continue doing that research to look at particularly what kind of bacteria are present and try to isolate those bacteria. So then we have strains of bacteria that are ready to be put in a starting new system whenever you need it. So those are the final results. The dominant bacteria that we have in the submerged biofilters for the recirculation aquaculture system, you see a lot of bacteria in comparison for the submerged biofilter that was work in the aquaponics. There was the most dominant bacteria were these four in the aquaponics submerged biofilter, but in the recirculation aquaculture system, there were more. Same thing if you look for the trickling or cascade type biofilter. There are more bacteria in comparison with the cascade or trickling biofilter working with the aquaponics. So those results, preliminary results that we have there, we don't have any answer why we have more bacteria in one system and less in other. Maybe the plants, the root of the plants are releasing some kind of antibiotic or are keeping some kind of strains of bacteria that we don't see in our recirculation aquaculture system because we don't have the plants in there. So I don't know, we are just hypothesizing some ideas, but we don't have any clear answer to that. We have now a project that we just started this year that we are focusing on to answer that kind of questions. And finally, just very quickly to close this speech, just to show you what we have doing with the high schools, where we are training the professors, we are also training the students, and we are releasing this technology that we have been studying in the university straight to the society. Here is Dr. Constanza Hanna from India, teaching the professor and students how to transplant the plants in the styrofoam bed before they move it into the hydroponic bed. With that, we expect that we are promoting social sustainability, teaching students and professors that are very connected to the society how to do aquaponics, and maybe they will be the entrepreneurs of this kind of technology pretty soon here in Chile. The system that we deploy in the schools are allowing the students to experience what they are doing, to share the result among the schools that are participating with us, and not just to share the result only with their classmates, but also to share their result among the other classmates in other schools. Also to understand the process that is going on in there, the process of growing the plants, the process of growing the fish, and also understanding the presence of the bacteria. They then generalize. They can generalize just by comparison what they are having in comparison with the result from the other places or the other schools. And finally, they apply this several times a year because the lettuce plant and harvest is growing on every month. So they are repeating and repeating several times and learning how to do that. So there are plenty of pictures and situations of us doing classes with the students, doing practices of moving fish, or in here that we are seedling with bacteria the biofilter to start up a biofilter, or the student uploading or downloading the fish from the truck, or a professor here just preparing all this system to read the water quality of the system. All this information you can be able to find out in our webpage from our project. And basically, are all the colleagues that are working here in Chile. From the Universidad Católica del Norte, you already met Maria Cristina, who is doing some of the research. From India, Dr. Constanza Hanna, and also some foreign companies. Bofish is a aquaponic company from Mexico, and Entorno is another company from Colombia that are also helping us to improve what we're doing and to release the technology. We have a lot of projects in the past with different topics with several institutions, and also we have a network, what we call it Civiolat. And in that network, we have these two web pages that you can visit, and you can see a lot of information in there, including the seminar that we are having right now. And of course, we have this other international SDG network that is responsible of the seminar that we are having right now. And for that one, we have this other web page. Please go to visit this one. In this case, this web page is in English. The other web page that I told you before is in Spanish. But please, both to visit the web pages and get whatever you can get in there. If you have questions, please do the question to us. We'll be very happy to answer. So that was all my speech for today. I'm ready to answer questions if you have any, Otherwise, I will say goodbye and have a nice day. All right. That will be all. As usual, I will say, Dr. Burgos, do you have any questions?
Yes, a lot of questions now. Yes, this is a good issue, and my congratulations for the presentation. It's very nice, very nice. You are an incredible professor. There is one of was your former student in this area. This is very excellent. It's very excellent conference. But now my question is about have you maybe some person, some researcher about your net have experience in tropical fishes?
Yes. And when I mentioned that we were working with international companies, the last ones, Bowfish and the other company, oh, I forgot the name right now. What a shame that I forgot that. Entorno. They are working with tilapia, barramundi as well.
Yeah,
So those would be, I would say, maybe more warmer fish. And also they have been, especially bowfish, they also were working, we were working with the river prawn, our river prawn that is from Chile and Ecuador and Chile and Peru only. But bowfish he was working with the macrobrachium rosenbergi. And in Brazil, they have been working with Lisa, there's a mullet. In Brazil, they have been working with tambaqui.
Yeah.
Yeah,
Now
that would
I
be
we
all I
we
would say.
were working with tambaqui now. Now
Yeah.
we here in Ecuador with in my
Yeah,
university
in
here in the
Brazil, in
Amazon
Belo
region.
Horizonte, they have already 16 aquaponics system deployed for experimental trials, and the fish will be tambaqui.
Mm, because this is the second question. Is it possible to participate with this net with you or maybe through you
Oh,
a, a
of course,
request of application?
everyone is invited to participate. You can. The only thing that you need to do is send us your request to be involved, and along with your request, a CV, your own CV.
Yeah.
And if you are a student, maybe also in your request, try to tell us, I would like to work, as you were saying right now, I would like to work with Tambaqui, who is working with Tambaqui. So I can tell you, oh, Dr. Ronald in la Universidad Federal de Minas Gerais, he's working with Tambaqui, and I can put you in touch, and then you can move over there.
Yeah, this is
Or,
very
or if you
nice.
want to do, I don't know, let's say you want to do bioenergetics, bioenergy or something like that, I can put you in touch with Dr. Rodrigo Poblete here in Chile, or with Dr. Camille or Guray in Turkey. Or if you would like to do other things related from the agriculture to aquaculture, I can move you to Norway. So it depends. So that is important when you write to us that you would like to be involved. Also, let us know beside your CV what area would you like to deep the knowledge so we can put you in touch with the right researchers.
Thank you a lot. And the other question is about your bacteria because you to work now in microbiota and microbiome in different parts of the RAS system, yeah, RAS and aquaponic system, yeah. And you have any comparison with fish, with intestinal microbiome and the different parts of the microbiome in the system? Because maybe there
No,
are a match for
yeah,
comparison. For example, I don't
no
know.
yet. No yet. In the slide that you're looking right now, here is Dr.
Uh-huh.
Christopher Concha. He's doing the microbiome studies right now. But also before, in our first seminar, I don't know if you were in the first seminar that was given by our partner from Norway, did you?
I didn't. I didn't there.
Okay, well, anyway, you go to our webpage and look for the first seminar, you will see the seminar for the partner from Norway. and one of them was also doing microbiome. And I guess they are doing it longer than we have done it. So maybe he has looked at the intestinal, water, and other places in the system that can tell you what is going on. Is the same or is different? Also, Doctora Maria Cristina, um, I don't know how many years ago, with another researchers, they were also looking at the microbiome in the river prawn that she was working with.
Yeah, because my thesis degree now for doctorate that now I have I sent my borrador my preliminary
Graphs.
thesis degree, my draft. Yeah, thank you so much. Because my director is Dr. Jaime Romero, maybe you know,
Mhm.
and we work with microbiota from Arapaima gigas. But
Uh-huh.
it's interesting to take the match. How about microbiome inside the aquaculture system, and what's happening inside with intestinal microbiome? There is so close in some cases, or so different in other cases. And in this case, in particular case of aquaponics and RAS systems, we have different knowledge that we need to learn because we don't know anything about. And there is a good question for try to put it together. And in my case here in Ecuador and the Amazon region in Ecuador, because this is a so far region from big cities here. This is what happened with the other materials that you use in RAS system. For example, um, uh, low cost system for us. This is the other question because the microbiome maybe in low cost material have problems with contamination. I don't know if it's possible because maybe
Yeah.
the low costs are recycled. Like recycled have maybe PFAS. There are very contaminants for the environment and for the fish too.
Yeah. Maybe so. There are another group that is also doing this research and that group belongs to the Aquaponic Association from the United States. So they are also doing the same studies in different aquaponics or commercial aquaponics that are spread all along the United States country. We started doing this because we found in our systems, basically in the aquaponic systems, that our fish never present any disease in comparisons with the recirculation system where we are able to see some diseases. So the people that work with plants in INEA, here in Chile, they told us that the veggies are able to produce antibiotic in their roots as a defense against the environment. So we are thinking that maybe in aquaponics, those veggies are producing some kind of antibiotics that maybe are moving around the whole system. And those are also maybe doing some kind of selection, what kind of bacteria could be and what kind of bacteria could not be.
And
That's
maybe
another
you
question.
have any identify what molecules, flavonoids or alkaloids or whatever molecules that are there
Yeah.
in these veggies?
No, we don't know yet. We are just, as I said, we are just starting this and we just opened the door and we are just started looking at it. So it's if you are working on the area, I will tell you, okay, you have a site where there is no path, meaning that whatever you do will be the path for everyone to follow.
Yeah.
Okay? So it's a very,
Thank you
very
so much.
interesting.
This is
No
a
problem.
values information for me. Thank you so much.
Yeah, no problem at all. Does anyone have any other question? Spanish or English will be fine. No problem with that. Well, if we have no more questions, I will let you go. It's right now in Chile, 11:30, about 11:30 a.m. It will be earlier in Ecuador, a 2 hour earlier, I guess, but it will be late in Turkey and Norway as well. So, well, I appreciate your time to be in this seminar. Thank you very much. For the people that have been in the 3 seminars, if you would like to have some kind of certificate or your participation, please let me know and I can prepare that certificate for you so you have it for your CV. All right. So see you for our next seminar and enjoy your day.
Thank you. Thank you a lot.
Okay. Goodbye.
Goodbye.